tyrosine phosphatase sigma A expression in the central nervous system of adult zebrafish and brainstem derived primary cultures. So let's draw your attention to this first image. This is an image adapted from a paper that was recently published in March about ptprsa, and this just shows the three-dimensional configuration of the binding site. So let's get a little background information on PTP first. So throughout this presentation, I'm going to be referring to it as ptprsa or PTP or PTP sigma because protein tyrosine phosphatase sigma A is quite a mouthful. So first of all, PTP is a receptor for CSPGs, and it's been shown that when it binds CSPGs, this results in an inhibitory effect on axon regeneration. The binding mechanism for CSPGs with PTP is a result of the gag chain. So if you remember, you have your CSPG with the protein core, and then you have the sugar chains, and what actually binds between PTP and the CSPGs is the gag chains. So here, what we have is a schematic of PTP sigma and how it would look. This was from a program called Ensemble, where you plug in the genetic sequence and it gives you a proposed structure based upon several key things that they look at. So here we have on the outside, we have outside of the cell, we have immunoglobulin domains. We have three. We have between 5 and 8 fibronectin type 3 domains. This is your transmembrane region, and then you have the two phosphatase domains within the cell. So before I talk about my experiments and my results, what I'm going to be doing is I'm going to relate to you, we've heard about L1, we've heard about Neurocan, and now we're going to hear about PTP sigma. So I want to talk, so we have a protein L1, a cell adhesion molecule. We have Neurocan, which is a CSPG, also expressed in humans, rats, and fish in the central nervous system. And now we have the receptor for CSPGs, we have PTP. So I'm going to present to you 4 models: non-regenerative, regenerative, injured, and uninjured models. So here we have a non-regenerative model. This represents a growing axonal process. And here, this would be an axon or glial cell, and this is your regenerative axon here. So as you can see, this is a non-regenerative model. We would expect few, so the red lines are L1. This is little boxes are PTP sigma receptors, and NCAN is NCAN. So here, we know that L1 homophilic interactions promote axon regeneration. So we would expect in a non-regenerative model few L1 interactions, right? So few interactions that promote regeneration. And here we show inhibition due to PTP sigma Neurocan binding, as you can see here, and L1 Neurocan binding. So a non-regenerative model would have few homophilic interactions and higher interactions of the inhibitory kind with PTP sigma Neurocan and L1 Neurocan. Our next model is a regenerative model. So this also represents a growing axonal process of, in this case we're studying zebrafish, so the zebrafish. So here we have high L1.1 levels, and the higher L1.1 levels naturally result in more L1 interactions, more promotion of growth, and less L1 Neurocan interactions, less inhibition of growth. And as well, we have Neurocan binding PTP sigma, which yields an inhibitory effect. But overall, we would suggest that the increase in L1 levels and the increase in homophilic interactions and the decrease in these heterophilic interactions would overall yield a net regenerative result in your cell. So now we look at an uninjured model. So what we know so far from what we've seen in all these past presentations is that we have L1, and we have PTP sigma, and we have Neurocan. So in an uninjured axon, we propose that you would have the PTP sigma receptor, not binding Neurocan, and just very few interactions at all in the cell. Because it's uninjured, it's just performing its function as a neuron. So in our injured model, right, this is an injured model, injured axonal process of a fish, we propose that because there's regeneration, there would be an increase in an L1 homophilic interactions, decrease in this L1 Neurocan interactions, and there would be an unknown quantity of PTP sigma Neurocan interactions. And this would yield a net regenerative effect on your axon. So now we've kind of looked at those models, we kind of understand a little bit more. I'm going to be talking about the methods that I went to in order to test PTP sigma expression. So because I'm testing a gene expression, I use RT-PCR. Now, Harold already talked to you about RT-PCR, and this is in fact a figure that he used in his presentation, so I don't have to go into detail. But the tissue we tested was uninjured brain, injured brain, spinal cord, laminin growth permissive cultures, and CSPG coded cultures. So you can see there's uninjured brain and injured brain, but there's only uninjured spinal cord. We didn't test uninjured spinal cord because logistically there was a problem there. It took about an entire day, 6 people, 10 hours to dissect 100 spinal cords, and that only gave me enough for one experiment. So uninjured spinal cords give you a significantly smaller amount of tissue, and so it would require 8 people, maybe 4 weeks to get about 1,000 spinal cords. And obviously there would be a logistical problem there. So using RT-PCR, now let's discuss my results here. So uninjured brain. So as we can see in uninjured brain, here we have the PTP sigma expression here, and then we have our beta-actin band, which as Harold discussed is our positive control. Now, when you read a gel, I would like to draw your attention to this minus column. This minus column is very important. It's a minus RT control, a minus reverse transcriptase. That allows us, that shows us that our, in addition to other controls, it shows us that our sample is uncontaminated, and that we do in fact have this. And sequencing has shown that we have indeed gotten this product. And so I've labeled the 500 base pairs. The band we're looking for is between 300 and 400 for PTP sigma and closer to 400 for beta-actin. So in injured brain, as you can see, we also have PTP sigma expression here, and this is our positive control beta-actin, and the minus RT columns are clear. The bottom, I know it looks a little bit messy, but the bottom is the result of excess primers and excess reaction mixtures. It doesn't really affect the reaction mixture. Then we have spinal cord PTP, and as you can see, spinal cord also expresses, this is uninjured spinal cord, expresses PTP sigma, and spinal cord also expresses the beta-actin. Then we have the laminin cultures, and as you can see as well in the laminin cultures, we have the PTP sigma expression, and we have the beta-actin expression. Then we have CSPG cultures. Now the difference between laminin and CSPG cultures is the coding of the slides. Laminin is a growth permissive molecule that promotes growth of the neurons. And CSPG cultures is a growth inhibitory molecule. So you would expect, so it's different. So we thought, the reason there was a difference there is because we thought, well, just in case the PTP sigma isn't expressed in the laminin cultures, maybe the presence of CSPGs would spark an increase in that expression. Now, it didn't happen to be true because, as we can see, laminin cultures also express PTP, as well as the CSPG cultures. So, our conclusion right now is that we know, according to Becker and according to our own research, that some axons are regenerative while others aren't. But PTP sigma is expressed in uninjured and injured tissues, and it's expressed in vitro and in vivo. So apparently, these types of culture conditions, or whether in vivo or in vitro, uninjured or injured, doesn't have an effect, or apparently doesn't seem to have no effect on PTP sigma expression. So now we know what happens and the interactions between NeuroCan, between L1, and between PTP. And so what does my results have on our proposed models, our regenerative versus non-regenerative models, and our uninjured versus injured models? So if we look at this non-regenerative model and we look at this regenerative model, we notice a few things. In a non-regenerative model, we propose that L1 would be expressed at a lower level, because if there's less L1-L1 interactions, there's less of a promotion of regeneration, and therefore there would be an increase in NeuroCan and L1 interactions. So a regenerative model, we propose that L1 expression is greatly increased in regenerative models. As you can see here, there's representative 1 interaction here, whereas you go to 5 interactions here. Now this is in no way a quantitative study of the interactions, but this is just a good example to show you what I'm getting at. Okay, so then uninjured versus injured. So an uninjured axon would be an axon that is simply in its physiological state. There is no outside factors besides what normally happens in your cell. Whereas in an injured state, all of a sudden you have things that need to happen. You have increased L1 interactions and decreased L1 NeuroCan interactions. So we propose that in an injured model, L1 is also, this is, I'm sorry, I didn't specify, but this is injured, uninjured, and regenerative and non-regenerative for zebrafish purposes only. Obviously, it would be a much different story in mammals. So we proposed that L1 would be greatly increased in injured models, and that PTP sigma would be expressed at a lower level because the the promotion of regeneration needs to overcome the inhibitory effects of PTP binding neurocan and of L1 binding neurocan. So now we're going to kind of discuss our results. So where does this leave us? So RT-PCR, right, tests only gene expression. It doesn't test protein expression. So I know that my gene is being transcribed into RNA, but I do not know whether my protein is being expressed, whether the protein is present, so I don't know whether my protein is present. And I also don't know whether it is present, but it might be inactive. And as well, so these are all, and the PTP receptor binds GAG chains, as we discussed in the beginning. It also binds GAG chains on other molecules called HSPGs and other molecules. So it could be that the PTP receptor is present, but is binding other substrates. So all of these could account for the regeneration of, or the regenerative capacity of zebrafish axons. So it could be that the protein PTP sigma is not expressed. If it's not expressed, then it can't bind neurocan. If it can't bind neurocan, then there's no inhibitory effect. Or post-transcriptional modification. Post-translational modification. The protein is expressed and it's there, but it's inactive. If it's inactive, it can't bind neurocan, and if it can't bind neurocan, there is no inhibitory effect. Or the PTP receptor is being blocked by binding to other molecules like HSPGs or other molecules with these GAG chains. So future directions. What we want to do is we want to experimentally determine whether this PTP activity is regulated through a post-translational mechanism or transcriptional mechanism, and determine whether this PTP biochemically interacts with neurocan in zebrafish. I would like to thank Dr. Plunkett and Dr. Alexis Stepanos-Castillo, and especially Francilithia Sabaj, all for putting up with me throughout the summer. And definitely I would like to give special thanks to Megan Studemeyer, Harold Gomez, and Gabriel Pacaro, who worked with me on this, and also Ma'am Moj. And anybody else I haven't mentioned, it's not because I don't thank you, it's just because I didn't have the opportunity and pleasure to be working with you over the summer. And especially to the U.S. Department of Defense and the Minority Science and Engineering Program grant for funding us and making this possible.
Questions?
Dr. Mall.
It was so good that they just kept applauding now.
So obviously you would want to quantitate gene expression and protein expression.
Yes, definitely.
Would you know how to go about doing those?
To quantify, well, a good real-time PCR would definitely be a huge asset in this case, being able to perform real-time PCR to try and test, I guess, to quantify the results of that RNA expression. Protein expression would be a different matter. I don't know quite how to quantify that result, but there's methods. So that's just me requiring to do a little bit more research into looking into that. Brother Kassan?
Okay, you presented in one of your slides a model where you have a different concentration of L1, the high level, the low level. Yes? It's a model for regenerative and non-regenerative.
Yes.
Okay, my question is, what's the mechanism that makes the cell to have that imbalance in the concentration of the L1.1? So one in some areas, or one in some neurons, you have a high level, one in some you have a low level, if you're covered from the same organism. So what makes that difference? From where that imbalance is coming from?
Well, that has to do with, that's a very, very broad question. That has to do with just intrinsic gene factors in each cell. You know, whatever turns on that gene or turns on that expression pattern, that has to do with, let's say, I mean, It's a very difficult question. Let me think for a second. Basically, it's within each cell. It's an intrinsic factor. That's very outside the scope of my knowledge, I think, because some cells obviously express a higher L1 level and some don't, but that has to do with more what happens, how the injury occurs, different types of biochemical molecules that interact. You know, if it, like say it like totally just throwing this out there, but if a molecule promotes this gene expression, some neurons might interact with it and some might not. That would yield to a difference in this expression. Or maybe some cells have this function already programmed within them and some don't, and that would result in this function. You know, there's many different types of cell types. So that question I can't really answer because there's so much involved in that, and this is so outside my area of research that's kind of beyond my scope to answer. I think that's kind of the question, you know, why, you know, of spinal cord injury in general. Why can't these, you know, that's what we're trying to answer basically. And the answer is that the research isn't fully complete and we haven't cured spinal cord injury, so I can't answer that. Yes?
I noticed in one of your slides you had an HSPG. What does the HS stand for?
Oh, heparin sulfate program.
Heparin sulfate, okay.
Mm-hmm.
Any other questions? Thank you so much.
Yeah, the question.
Oh, sorry. Oh, sorry. Excuse me. Because in the same slide you mentioned that, of course, it's a zebrafish model. Right?
Yes.
And then you said that this would be very different from a natal model, of course.
Mm-hmm.
I'm not sure what you meant by that, because the ultimate idea is to cure spinal cord injury in humans.
Yes.
So how are we translating the such model into at least in a
Mm-hmm. Exactly. Very, very good question. Well, first of all of these molecules have close homologs in humans. Secondly, this avenue of research is more oriented towards curing. Let's say you get into a car accident and your spine is partially crushed and you have an injury there. It would be curing those trauma cases because it's a much different area of research and of whole methodology to treat a spinal cord that's been injured for 14 years. So this would be for immediate sort of treatment. And secondly, knowing how these molecules interact with each other and this sort of interplay between these molecules would help give us that treatment for an immediate case. Like, so for say somebody in that scenario gets injured and we know what molecules interplay in a specific way in order that spinal cord doesn't regenerate. Well, if we could find a way to inhibit those molecules or to promote growth permissive molecules or promote regeneration in such a way that doesn't interrupt physiological function, then we can then that would allow us for that patient to actually regenerate those axons. So I guess what I'm trying to say is that, for instance, CSPGs can be treated with a molecule, chondroitinase, that cuts the GAG chains, right? So when you cut the GAG chain, they don't bind PTP sigma, and then there's no inhibitory effect. PTP sigma can also be treated in order to keep it from binding CSPGs or to binding with those GAG chains. And so so once we know how the molecules interact and specifically what they do and how they affect axon regeneration, then we can take it to a mammalian system. So say we know what happens in zebrafish, we can take it to rats and then try and treat it in rats. So you perform an injury and then you put in the correct correct molecules to try and influence that area and influence the conditions in that area, and then therefore promote the axon regeneration. So it's not, although it's just much easier to study in a zebrafish.
So models,
Yes. Any other questions?
Thank you.